Hello, everybody. Welcome on in to another edition of the GSA on Twitch. My name is Balint, and today we have a very special guest star. Kaushal Bhati is here with us, former early career scientist with the Genetic Society of America and plant geneticist extraordinaire. Kaushal, welcome on in. We're very glad to have you on today. Would you mind introducing yourself and telling us a little bit about where you work now?
I'm Kaushal. I'm based in Ghent. Ghent is a small city in Belgium, Flanders region, and I'm working as a scientist at BSF Innovation Center. BSF deals with a lot of different businesses, but I'm based as a scientist in trade research, and we do research and development for engineering the new traits in seed, especially some commercial crops, and target regions are North America, South America, and Europe. So it's a mix between basic fundamental research discovery as well as delivering the applied biology, applied science in terms of plant biology and seed biology.
And so it's much more in the pharmaceutical side of things now. Is that correct?
It's on agriculture side.
And so what, like in the current route, like we haven't had anyone who's working on like this agricultural science yet. So
Uh,
what is your day to day like? What is the mission statement? Because that's very different than working like in an academic setting. So how does something
yeah,
like that work? What do you decide? Do you decide to study what you want? Are you given a project to do? Do you take it to completion? Is there any moment do you switch projects? Like how does that
mm.
world work?
It, it's quite mixed. Based on your role, you do decide what you want to pursue. But mostly, in my role personally, I work on multiple projects. The project could be about AI integration in your business, but at the same time, you will also deal with assay being optimized for let's say some expression study or some assay to detect genome edited plants if editing has happened. So there are tasks being given to you. Those are needed to complete a bigger project, and you allocate to project based on your expertise, and then you provide expert advice there, or you talk about new developments or the new needs that project might need to finish at the time, or on research investigation part, I contribute to development of new technology, new assays, new methodology to deliver something more quicker, better, and faster. So
What
it's
has
quite mixed. Yeah.
that's really cool. You mentioned genetic engineered plants as well that you're currently
Mhm.
focusing on. What kind
Yeah.
of plants are these? More like things that you would be eating, or or can you tell us? I suppose like what not necessarily a particular
Yeah.
plant, but like a group of plant. Like is the overarching goal to make things more
Mhm.
growable in different regions of the world, more resistant to disease, maybe a mix of both.
It's mostly a mix of both, but these are business strategies. Those we don't decide really. It comes from top. It's not top-down approach where business decides we need these kind of new traits in market, considering the climate change, then new challenges coming up because our pathogens are evolving really quick with the climate change as well as the genetic drift. So, as a scientist, you need to you need to cater the need for future. You don't just look for short term, but as a business, you look even further away. You look for fact that at some point the crops you were growing or you were delivering seeds, they might get out of market because of some resistance evolution or development of new genetic drift in one of the pathogen, and then you are out of market again. So these are really uh, farmer oriented approaches. They are a bit different than the fundamental science that I used to do in past.
Because of how it's designed, right? Like that long term,
Absolutely.
even longer
Yeah.
term. And you
Yeah.
mentioned the use
Absolutely.
of an accounting for climate change. So
Mhm.
I'm very fascinated by that topic. How does one develop these like new genetic tools in plants while taking into account climate change? Like how do you, what kind of models do you use to predict climate change and what feature of the climate change are you really looking for to
Mhm.
alter in the plant? Is it just simple as heat resistance? Is it carbon dioxide content? Is it how does
Mhm.
one go about that type of experiment?
I can't be very specific in terms of my employer and background here, but in general, I could speak probably for community, the plant biologist community. So there is a really good example that the yield loss that Europe has experienced in past 2, 3 years because of extended heat waves and less rain actually. So what it causes reduce the overall productivity for crops as well as the quality of grain is actually deteriorating. It's not enough protein. The quality of protein in grain is decreasing, overall yield is decreasing. And then again, if there is no rain, and then farmers those are not using artificial irrigation, they want to have standing crops. Those are resistant to the extended drought, which you can't really foresee. So approaches are again approach mostly drive from fundamental discoveries. For instance, if you identify an aquaporin, and aquaporin is a transmembrane protein which channels and mediate homeostasis. It's just a figurative situation which I'm taking here. And you study this aquaporin for 12 years, 15 years, and then at some point you realize now is the time to translate the knowledge we developed during those years into application, for instance. And then you start engineering your plants by expressing those aquaporin in roots, maybe in one of the crop which is more threatened. And then you start field trials. You see when you express specifically in root, do you see more water absorption? Do you either see more retain of water, more moisture? And these are like approaches those who evolve from fundamental science, understanding at cellular level, tissue level, and then to organism level or a crop level. And starting from this fundamental to delivery, there are several steps which, as an academic researcher, you are not really aware of, but you realize those big regulatory frameworks and the limitations you have on use of components to drive a gene and IP regulations specifically, which you really learn when you start working in a big company because now you are dealing with something will definitely go to a field trial.
Yeah,
So
that seems like it
it's
would
a
be
big
a
transition.
it's a
Yeah,
different world, right, than in the academic sphere,
exactly. Yeah, yeah,
which we'll get
yeah.
into in a little bit. But so, Kaushal, I'll take us back. Like, why plants? What got you interested in plants to begin with, and like, what was the like your first experience with research?
Mhm. So I come from a farming family. My parents, we actually inheritedly involved in farming since I don't know every generation maybe. So when I was growing up, I was growing up with parents or family those were continually involved in farming. I worked on farm with my grandfather, my father, uncle, and I intensively contributed to farming. So it's like taking everything in hand or helping all of them at some point with farm, fields, animals. We have cows in family.
What kind of farming was it?
So,
Like, what were the crops and output?
so at that point, we were growing a lot of corn, rice, wheat is one of our major that should grow every year because we consume our own wheat, which we are growing on field. And I also remember vegetables like chili, and we had this mix agroforestry concept, growing chili with the cotton. So I have really good memories of running through the cotton field because it looks like a maze, and then you can play hide and seek while everyone is working on the field. And yeah, while growing up, I got interested more into understanding the biology, but I was not really inclined to plant until I started some courses in tissue culture in plant during my bachelor's, and which really excited me to regenerate something from like really small X plant, you can regenerate whole plant through micropropagation and in vitro culture. And even till then, I was more interested in immunology. But at some point, I had to make decision when I was moving to university that now is the time. Do I like immunology more than studying plants? So I had this master's in plant science then, and it was definitive that I will study plants now onwards.
What was the influence? Like, how did you know plant science over immunology?
So I think influence was more on that you can really see plants growing, adapting to the change, being more plastic. Uh, Plasticity is one of the most key feature which I don't like in plant. For example, you don't. I mean, I grow a lot of indoor plants now. So if you don't water them for 2 days, you see those changes. Like they are really apparent. And then you suddenly water, and next day they are happy again, smiling, laughing at you. So I think it's more like what you see, you spend more time with, you get more attracted to. Immunology is really good to read, study, and see those interactions between cells and overall physiology change. But plants you can really see. So while on the go, there is a concept called plant blindness, which most people have. I was really aware of everything what was happening around me in terms of plants. So it was like a more natural inclination.
Gotcha. So you could go
Yeah,
from working in a dish to the actual plant,
yeah,
and then back to the
yeah,
dish, and you can see like a physical change versus just looking in the
yeah,
dish
definitely.
only. Yep.
Yeah, yeah,
That my interest in
yeah.
insects is the same way. It's like you can see that cross like on the organismal level, and then back to the molecule and
Yeah,
back to the organism.
yeah.
So then you
Yes.
said you got your
And
master's. So
mhm.
where and that was in the plant genetics. Where did you do that?
I did masters from Banaras Hindu University. It's a biggest university in India in holy city called Banaras. It's also one of the touristic place and oldest city, we call it oldest cultural city that represent Hinduism, let's say. So the university itself called Banaras Hindu University. So yeah, I did masters there. So I relocated from my native place to Banaras. I spent almost 2.5 years there, and it was in agriculture faculty. It was quite fun time because that was the first exposure to really professors who were working with farmers, doing the breeding, making those all genetic changes that was ultimately going to benefit the farmers, which was really inspiring actually.
So you worked with the farmers directly, like if you found some kind of result in the lab, you try to translate it and help
Yeah,
the farmers directly, and then they give you
exactly.
feedback.
Yeah,
That's really
right,
cool. So
right.
you
Lot of
got to be
yeah.
on the front line of like difference making research in people's lives. That's
Mhm.
very unique, and most
So
people don't ever experience
yeah,
that.
yeah. It's actually a privilege only breeders have in breeders specifically in like plant biology breeders, plant genetics. And in India, we have a big network of institutions where breeders directly interact with the farmers. They also contribute to seed distribution, scoring how certain genotypes and germplasm are doing during the farming time or harvest time across the different geographical location. So it's very supportive environment, I would say, and which makes you think that in that fundamental biology is one thing, but also translating to what really going to in hand of farmers, it's a different pleasure.
How did that
Yeah.
feel?
I mean, coming from farming family, it's a different feeling, of course, because I always heard as a young student the breeder, the plant scientist, or the scientist, but we never met any while growing up because the network of breeders it's only limited to let's say bigger farmers, those who grow big hectares and yeah. But university time was different, where you actually go to. We also have in India, it's called farm fairs. These are like carnivals, but on very small scale, where carnival is being organized by university, and then it actually invite farmers to the carnival, and then farmers has opportunity to uh, talk to scientists who are developing new technologies, but they can also talk to different industries. Those are coming here as a stall maybe. So these carnivals are totally dedicated to farmers. And from university perspective, as a student, you go there, you volunteer for these fairs, carnivals, and then you see how farmers are feeling about seeing something new coming into market or seeing a new seed that could be very revolutionizing for their farming practices or could actually enhance their earning. So yeah, it's a different feeling
That
than sitting in a lab.
that's really really cool. Did you get to give any scientific advice to like your family? Were they still doing the farming business, and like could you talk to them and help them as well?
Yeah, I tried actually. I tried because I mean, after finishing my master's, I did my PhD on wheat, Triticum aestivum, the bread wheat, and there also I learned a lot about growing wheat, but more in scientific way. So often I used to have discussion with my father about, for instance, length of flag leaf, how flag leaves are looking. So when these monocots, especially grasses, the 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 triticum family grasses, when they grow, right under the spike, it's a big leaf called the flag leaf. So I'm really fascinated by wheat, especially to understand how flag leaf contribute to grain development. So I was often asking my father when was the time to harvest, or let's say we started seeing the spike. I was asking how the flag leaf is looking. Are they undergoing senescence really quick, or when did the wheat flower? How was the flowering? Was it like all field at the same time, or was it starting gradually from one side of field to other side? So these are like interesting things which you see in lab, but now you want to explore them in your own farms.
Yeah, that's
Yeah.
and especially like that family connection. I feel like makes it all the more special.
Exactly. Yeah.
I
And
Kaushal, I love your story already. This is so cool to have that connection, like especially then
Mhm.
helping other farmers with the genetics during the masters, and that's connecting back to your
Yeah.
childhood. Like that's a beautiful
Mhm.
story already. Um,
Yeah.
what did so when you were getting your masters? What were you studying then, and like what kind of like what was the overall project that you were trying to identify in plants?
My master project was really it was really funny. So my lab where I did my master thesis, they were doing they were studying a leaf blight. It's a fusarium leaf blight pathogen for corn, and it was mostly breeding, finding genetic markers associated with the resistance in certain germplasm. And as a fun project, what I did, I I started cultivating. I extracted that blight from fungi, fusarium, and then we started cultivating that on plate. And in my university, we had a garden. It's a garden for native medicinal plants. So I contacted one of professor who was studying those medicinal plants, and we started collaboration thinking that could we really use plant extract to control the fungi? And these are native Indian plants, so he advised me to use certain XXXS plants. And what we did is we prepared different extract in let's say alcoholic extract or the extract in simple water, high aquatic extract,
Naive
and then we
question
start
for you, Kaushal.
dipping.
What is an extract of a plant? My brain just imagines you put a plant into a blender and you drop that onto a dish.
Exactly.
Is that what that
Yeah,
is?
yeah, yeah.
Okay.
Yeah, you crush that in a solvent, the leaf, and then when you remove after centrifugation all the debris, whatever you get out of it, You use that as a concentrate and drop it on plate. It's called inhibition assays.
Okay, cool.
Yeah, yeah. So you grow fungi on plate, you inoculate at the same time fungi as well as your drops of your inoculum, the extract inoculum, and then you score how your fungal is growing in presence of those extract which you applied through the plate. And then you start seeing the inhibition curve that around the circumference till this extract was spreaded, you don't see any fungal growth.
Okay.
Yeah, it's
Wow.
it's like a very basic, but at that stage, it was really interesting to think those, yeah, those approaches. I
So
mean, for master's level, it was already very exciting for me.
no, that sounds so. Why was there growth in some instances and not in others?
I think all because of phenolics, because some of these medicinal plants they are really high in phenolic content,
Hmm.
and and then the overall phenolics, but maybe could also be other phytochemicals, those who inhibit hyphae growth in fungi. Yeah, so it could be how do we say allopathic, or there is a term to define these kind of plants, those who have this metabolic profile where you can, I mean, it's all metabolic metabolites. Those could be making those medicinal plant more resistant to fungal infection or pathogens. And when you prepare this extract and you apply extract on your inhibition assay plates, then you see impact.
That's really good. See, I would have naively thought maybe it would have been also like immune system components, like some kind of autophagy
Maybe,
that hit and starts like eating up the fungi or something along those lines.
no, no, I wouldn't go that way. Autophagy is quite cellular process. You can't. Uh, I mean, we can't really say because autophagy is really complicated. That's also one of the
I know you're an expert in
possibilities. Yeah,
it. So
yeah. It's
you're doing these assays, you're working with
mm,
farmers, you decide to go get your PhD next.
yeah.
So tell us what was that like? Where did you go? What was the big project, the question that you were trying to answer, and
Mm,
how was it different from the master's experience that you had?
yeah. So PhD in India is it's a different way of selecting your PhD. So first, after finishing your master's, uh, you apply for funding, and funding doesn't come to you through writing proposals or grant. You need to appear for a really competitive national exam, and if you qualify that national exam, then you get promised by funding agency that you will get funded when you start your PhD. So we have like two, three layer of selection. If you want to go to the best institute in India, first you qualify for national exam, you get the assurance that you will get funded. Then you start applying to these big institutes to say, I want to do PhD at your institute. An institute will shortlist you then for your scores in masters, bachelors, and school. And then they also ask you to appear for interview again for after qualifying this cutthroat competition for fellowship, then you appear for interview again. Then they make a score based on everything in your profile. And at some point they will say, now you can come and do your PhD. And once you start your PhD, then your funding agency will release your fund for 5 years.
Can you pick what lab, or are you interviewing with more of an institute?
Yeah. Yeah, you can always pick lab. But in some institute, it's a bit complicated. You apply for PhD, then you will be picked by a supervisor. And
Okay.
you wouldn't control which supervisor are you going to work with. That might bring the conflict of not having the right supervisor, right vibes being matched between two people, and, and that's a bit messy. But it's not common for every institute. It's just some institute, those who pick the candidates very randomly.
It seems like it can be really complicated to find a good fit, right? Because you're going through these exams and
Exactly.
all these loops, and you're only
Yeah,
getting that funding released if you identify a school that has now approved
it's good.
you
Yeah,
and you're accepted, right? And
exactly.
so what happens? And this maybe you don't know, but like if you want to change a lab, is that even on the table?
Just once in one funding. Otherwise, you appear for exam again and qualify it again, and then you can change with the second funding.
With more interviews again, right?
Yeah,
Wow.
exactly.
So that's that's quite a very different journey than in the U.S. where
Yeah,
there's
yeah.
not the quality. There's also no guarantee of funding, right? I guess that's the
Mm,
benefit, right? Is that you can have
mm,
funding guarantee for X number of years versus here,
yeah.
instances of losing funding for a lab and having to shift to a different lab can happen and
Mm.
does happen.
But but if you look at the candidate size, there are only like 800 candidates being funded for one of the funding agency, which covers all science parts. Then for biotechnology, we have a ministerial section. It's called Department Biotechnology. They have their own funding, which is only like 400 people being funded every year. And for this exam, I guess it's like 100K people appear for exam. And then in this really difficult competition, you appear among those 200, 400 in the top. So yeah, it's a different scale of
Yeah, it's a
study.
very very different world. We
Yeah,
have a question from chat like before like
yeah.
you're even starting at the university. Max is wondering why would you study something with more than two sets of chromosomes, and why do you like working with plant genomes versus animals?
Yeah, actually, I never, I never choose to know about difference between genome when I started loving plants because I was quite naive to say I will choose, let's say, uh, a hexaploid, for instance, instead of diploid, or I would choose a plant genome which is more complex and huge compared to animal genome. So it wasn't really choice based on what I'm going to study, but it was more like I like plants and I don't care what they come with.
So the genomic instability was like an after effect that you dealt with later
Exactly.
versus
Yeah. That was really shock when I started my PhD, as I mentioned, for it was in wheat, and the lab was really new. I was first PhD student for my boss, for my PhD boss, and before coming back to India, he was working with soybean and starting a lab, then working with a giant crop like bread wheat, which is hexaploid and one of the thickest and complex genome. It was big shock because I mean, personally, I never imagined how complicated things could be. And then you get to know that you can't really transform genetically transform bread wheat because it's a calcitrant crop you can't regenerate if you create a calai out of it. So it was a whole set of challenge. Then I saw my colleagues in different lab, those who were working with Arabidopsis. I'm like, why did I choose to study wheat instead of Arabidopsis? Which you can just casually dip in soup of agrobacterium and get it transformed.
right.
Well,
So is there?
uh,
Can you not do the transformation on your model organism, like during the PhD? Like,
I I did actually. I spent 3 years creating just one transgenic.
wow. What? So,
Yeah.
question for the audience, like from the presume the audience will also be interested. Why? Like, what's the difference that took so long? Because, like you said, for Arabidopsis to change a gene, it's honestly you just dip it in a mixture, right?
Yeah.
And then you have a
Yeah.
changed gene.
Right.
But like, why? Like this 3 year process, did you help? Did going through this arduous 3 year process help potential future researchers in this method? That maybe it would be easier for them, or is it just going to be like Kaushal? I want to change a gene too. Now I gotta put in my 3 years. Like, is it going to be like that every time?
No, the bigger purpose was institute where I was doing a PhD. It had a mandate to improve Indian wheat varieties, and then bigger and the biggest obstacle was there that you can't really genetically transform wheat because you need to validate your gene functions in plant itself. So my project objective was to identify a pathway, a metabolic pathway. It's called phytic acid biosynthesis pathway. So how it works is when your grain is being developed in wheat, you have an outer layer of single cell in grain. It's called alluron layer. And since India is a big wheat consumer, and it's very monotonous diet that we have, and most of us don't eat meat at all. So the source for iron for Indian vegetarian people, it's really difficult to get enough iron. So the bigger objective of institute was to develop different approaches so that we can increase iron either in grain or we can increase bioavailability of iron. Now, iron get trapped in a compound called phytic acid, which is the storage form of phytate in grain. And phytic acid can't be digested by us, monogastric animal, because we don't have enzyme called phytase. So by covalent interaction, iron, zinc, calcium, they got attached to phytic acid and then they don't get released in our gut.
So that would be the
So
increasing the bioavailability, right? That would be like
exactly.
as you're
Yeah.
trying to make it so that within the plant it's perhaps maybe broken down easier so that when we eat it it's more
Yeah.
accessible, or
Exactly.
totally increasing the concentration. Or
Right.
maybe
Yeah.
first of all, that project is really cool. Like that is a such a cool, and again you see a direct application
Yeah. Yeah.
to the world, right? Because like you're saying, like I
Exactly.
know not just in India, but anywhere, vegetarianism like that has a low level of iron intake, and that's
Absolutely.
always like
Yeah.
anemia is a big issue. And so imagine how many
Mhm.
people you could help by doing that
Exactly.
change.
Yeah.
So
Yeah.
what did you end up doing to like what was the answer? Was it both? Was it one? Was it what did we do?
Still, there is no answer. I mean, I can't really comment on that part this early, but the story evolves further that we tried first to see how phytic acid is being synthesized because to reduce phytic acid, you first need to know how it's getting synthesized. And in crop like wheat, where there was no genome information available, so we started digging through microarray data. The modern generation might not know microarray, how precious was it at some point, and uh, and then we started developing a map how possibly phytic acid is being synthesized. Then
And
at the
just
same time,
Kaushal, just
we
to make sure I understood, you're not working with a genome sequence at this time.
no, it
Oh
was
wow,
it was just random hit because there was no genome sequence available.
wow,
Yeah.
that that
So
seems like a very difficult starting point.
exactly, and being a new lab, it's a whole different challenge because your PI, you guys have never worked with beat before, and now you need to work with this complex system. But luckily, the whole institute was working on beat, and there were some other projects running parallelly. For example, RNSec was new thing that time, so we get some early quantic library from RNA sequencing and those things. So we started annotating genes for phytic acid biosynthesis, which was first paper from my PhD. And then seeing other crops, how other people are reducing phytic acid, let's say in corn or rice. So we started developing this approach, saying like, how are we going to reduce phytic acid? So my target was mostly on transport part. Once phytic acid is being synthesized, it gets transported into vacuole through ABCC transporter. Then we developed a whole genetic map of ABCC transporter in wheat genome, and then suddenly there was actually genome published around 2014 something, but like partial genome. These were early assemblies and contigs. So we were quite lucky. Then I started cloning gene and the complexity of genome itself. Then the gene cloning, gene size was like 4.2 kb, and amplifying that big stretch from read genome, genomic DNA it was really a big challenge. Yeah, so then we further evolved. We started studying more, and then at some point we decided, the project decided that we should silence the transporter so that we reduce transcript for transporter. and there should be less protein and less transportation of phytic acid into the vacuole, the storage vacuole. So that was the time when I got struck by reality that to silence this gene, I need to genetically transform the wheat using RNA, and which has never been done, at least in our institute. So I started understanding with other colleagues, those who were already trying those transformation approaches. And after spending 3 years, why 3 years is because you start your experiment in a batch, and then suddenly you realize at some point that all calli you prepared were contaminated. So they will new germinate. Then you throw that batch and start the next batch again. So it's like every 3 months you dissect the calli, dissect the immature embryo, then you put immature embryo on a plate to make a calli, and then you transform them with agrobacterium. And if that batch doesn't work, it goes into waste, then you start next batch. So I did like 6, 7 batches,
How
and
many
at some
embryos are in a batch? I'm just like wondering numbers here.
500, yeah, 500 or at least 500, yeah,
So 500 times, so over almost 2,500, if not
exactly,
even more,
yeah,
thousands. It's
yeah.
wow. How many? So how
The,
many
the
did you
job,
end up with that were transformed of those? Let's say 5,000.
uh, yeah, in my last batch, where I really got the transgenics, it was like 5 or 6 transgenics.
5 or 6
And
transgenics out of several thousand.
exactly.
Wow.
And in between that, there was one incident happened that I had a batch running for transformation, and then that was collided with my wedding date. So I took a week off or something. I went to my wedding, and then on the wedding day, one of my student was calling me. She was helping me with transmission, saying, "Oh, we got contamination in your plates. What should I do about it?" So I asked her, "Why don't you go and talk to someone else? I'm really kind of involved in something now." So she went to someone, and then the person advised her, "You can't keep contaminated plate. Why don't you throw them all?" So I asked her to click picture. She sent me the picture, and I realized they could have been survived even after contamination. But she immediately threw all the plates. When I returned from my vacations, I asked her like, "Did you manage to subculture those calli again?" And she was like, "No, I throw everything away. I was sure that they would survive." So that was one of the promising batch which went into the bin for for my fault that I was not there to make a decision.
How was the
Yeah,
wedding?
it went well.
Good, good. That's I would be so
Yeah, yeah.
stressed out. You get a phone call, you're away on your wedding. Hey, everything looks like it's gonna die. You kidding me? No. And then you get back. Where is everything? Oh, it's in the trash. That's fine.
Yeah, it's all in trash. Yeah, yeah, it was quite frustrating because you worked very hard on dissecting, then subculturing. It's like a really laborious work. Yeah.
Yeah, but it makes for a good story, right? At least we're smiling now.
Yeah, yeah, exactly.
So, when you got your mutant? Did you find that there
Mhm.
was a difference? Like, was your hypothesis proven correct with that transporter, or was it
Yeah.
something different?
So me and one of my colleague in group, we took two different targets. I took the transporter, she took one of the biosynthesis gene. And when we silenced, we started doing metabolomics at grain level, and we saw reduced phytic acid. And we also, this was one of my PhD paper too. So where we see silencing was actually because wheat is a hexaploid, so every gene presents 3 times. Actually, 6 copies, but 3 times if you consider the. So the gene I was targeting was also in 6 copies.
So you had to mutate
So
all 6.
exactly. So you need to silence either all of 6, or you need to at least say you were able to silence some of them. So the silencing was like 43% something because we did some real-time PCR, and we quantify individual allele for each genome. And then we saw that all genes were noticeably silenced. So the reduction in phytic acid was only 36% because of buffering. That not all transcripts are silenced, but some transcripts are still buffering the activity, resulting into hyperaccumulation or let's say better accumulation.
So it's like
But
there's genetic backups, right? Like if you break one, there's
uh,
others, and they might, you don't know if they even will
yeah,
compensate fully, right? Which is why you have to do the measurements and see.
yeah, definitely. Yeah. So that's the complication. And the question, which was like, why did I choose the hexaploid plant to study? That's the answer. I didn't really know.
It's yeah, it
Yeah,
sounds more and more like, oh my god, there's so many
yeah,
variables.
exactly. Yeah. And at that point, while I was wrapping up my PhD thesis, I realized it's not easy to work with weed. You can't clone a gene, and it was not the time where you can just ask the companies to synthesize a fragment for you. It was really costly back then. And coming from India, where we always struggle for resources, especially everything is so costly in biological science. So you need to spend at least 1 year cloning one gene, which has now changed because you don't clone gene anymore using PCR. You just order the fragments, you assemble them, and there you have everything ready.
So
Yeah,
today, if you were to redo it today, it would be
yeah.
easy. Even though it's hexaploid, it would be easier and faster, presumably.
Far easier, far easier. And now there are better protocols for retransformation using the morphogenes. These are called growth factors. So if you express growth factor in your calli actually regenerate very quick. So in my current institute, when I was talking to one of the wheat transformation expert as an introduction, I felt like humiliated because they have 90%, 99% transformation efficiency for wheat.
Wow.
Yeah. So what he was saying, it was really funny that we just touch the wheat and it gets transformed. I spent like 3 years of my life in transforming the wheat and get only like 5, 6 transformations out of it. Yeah.
And now it's just like a
So
fast.
exactly. Now there are better protocols, better approaches using morphogenes or transcription factors, those who really change the overall transcript scenario and regenerate things very quicker.
Very cool. Very cool. So,
Yeah,
yeah, well, in the chats, it's character building, right? You had to go.
yeah, exactly. Yeah,
So,
it's a castle building. Yes.
with that reduction, you said it like reduced 36%. That transported, did you find the answer you were looking for using that mutant?
Exactly. The down the lane assays were to use cell lines. These are called, these are special cell line to study iron uptake. So it's called Kakatu cell lines. And when I finished my PhD, the project objective was to test those grails we created in those cell lines, and which was the time when I already left PhD. Yeah. So my PhD objective only was till reducing the content in grain, phytic acid content, and then it was someone else work to test those grains.
Gotcha. So it's almost like a
Yeah,
continuation. It's not necessarily that it
exactly.
like a
Yeah,
full bow on the project and like a present done.
yeah,
It's
yeah, yeah.
building on each
It's a
other's.
big ambitious project from government of India where I contributed just a tiny part.
Well, I would say a
Yeah.
very big part. So,
Yeah.
with that, Kaushal, you're now married too,
Mhm.
right? As you talked about with one of the transformations during the wedding date, you decided to go do a postdoc. So,
Mhm.
I think one question that we commonly get is, first of all, what was like, how did you choose that postdoc and what was it in? And then if it's on a similar field, like for example, plants, what drew you to stay with plants? I know it's going to be a different topic,
Mhm.
but if you could weave that
Mhm.
into your answer, why you were looking for a similar organism, from grad
Yeah.
school, masters, now
Mhm.
to your postdoc, and how did you select and
Yeah.
where did you go? And
Mhm. Yeah. Actually, this character building during PhD, it really helps because my supervisor, PhD supervisor, he was really cool. I mean, he's still very cool. So He really helped me in seeing science in a different way, which I'm really grateful for because we had really cool discussions, just not about our project, but overall how world is changing in terms of science. And at some point, he was organizing a conference. It's called Arabdopsis India, where people from who are working as an Arabdopsis model, they usually come together and they present their work and discuss how they can collaborate together. And most of them are developmental biologists, those who study stem cell niche or plant plasticity in Arabidopsis or flowering time and those things. So that conference was really striking for me because I was struggling with weed. Well, I had very defined objective, which I felt if I achieve that, I will be really grateful. And then I saw suddenly that people are doing really cool imaging. They are doing chipsack, they are doing pull downs using Arabidopsis as a model. So that was the point I decided that I will work on Naruto now onwards to really experience the fundamental science because you can't do those things in crops like wheat or soybean. It's really different and very challenging. But at the same time, I also got advice from some senior people saying you should spend time on wheat or specifically, let's say, evolving as a wheat guy because I was the first one to transform wheat, then the same protocol replicated again by other colleague. So I felt like, yeah, I want to probably keep working in wheat, but this conference really changed my perspective, and I felt I want to experience more. I don't want to limit my science exposure only to wheat, although it's really cool and challenging, but you can't do chipsack, you can't do flowering assay, or you can't look at the stem cell niche in wheat using different markers or fluorescent dyes or those things. So I also, during this maturation of PhD period, I started realizing that if you want to really become a scientist and feel like a scientist, you should get trained in different parts. So learning how to transform wheat, how to do metabolomics or study seed development was one part, but how would you get a chance to study flowering time, for instance? And that was the time I started looking thoroughly on advertisements, what kind of positions are being offered, what are the projects. And it was my first application I sent to Stephen in Copenhagen because it really excited me to see that there are new concept called microprotein where a protein goes and silence another protein or inhibits its function. And I was already working on RNAi in wheat, which I really find the microprotein concept itself, I really find it exciting. So I saw it on Twitter. That's why I
Twitter.
also tell a lot of
Okay.
people to use Twitter. Yeah. So I was writing my thesis. I sent this email to Stephen saying that I have experience on RNAi. I was already helping my boss in developing CRISPR-Cas9 construct that time. And that was the boom when whole world was shifting their focus from RNAi to CRISPR. And then no one was really doing the RNA-seq anymore. They were all doing more better techniques. Or microarray was actually going down. RNA-seq was more routine. Everyone was doing RNA-seq. So it was kind of transition, same transition from not knowing the genome, having the genome, and now doing everything routinely. So I wrote email to Stephen and he interviewed me, which I really liked. It was my first exposure to a PI in Europe, and I felt very comfortable in conversation, in interview. Then I also got to talk to my colleagues, I mean, let's say future colleagues, which was also big culture shock because I got my PhD without even knowing whom I'm going to work with or what will be the project. And now suddenly I was making decision that these are my colleagues. Do I like them? That's my supervisor. Do I like him? And that's the project. Am I going to happy with the project? So that was only application I ever sent for postdoc, and it got accepted.
So it's a
So
very different path than how you applied for your PhD as well. I bet that there's some
exactly,
shock between
yeah,
those two, like just a
yeah, it
massive
was a big
difference.
shock. Yeah. So And also big shock that a lot of people, those who were finishing PhD at the same time, like me, they were sending hundreds of application. I only sent one, and that actually worked. So, and Stephen was really kind that he offered me position when I was writing my thesis. So I had like 4 months, finish your thesis, defend your PhD, and then come very relaxed. But but then life happened that my PhD evaluation got delayed somehow, and then I need to really rush through finishing everything to catch the flight. So I defended my PhD on 26th of August, and I had my flight to Copenhagen on 28th of August.
Oh no, no.
Yeah.
Wow.
So we packed our house. I was in different city then, and almost like 300 kilometers away from my parents' house. So we packed our house. I went to my PhD defense, and immediately we fit everything into a big truck and then drive back home. And the next day, I packed my bag to Copenhagen, and so it was like really
Wow. When did so you
uh,
when did you start work? Did you at least have a little buffer?
no buffer. I actually never been unemployed technically, except last couple of months before starting this job.
So it was you got off the plane in Copenhagen. You got a
Exactly. And then
place, presumably put your stuff in there, and the next day you're like off at work
yeah,
already.
I was in office already. Yeah, right.
So
Yes.
wow, that is Kaushal. It's really stressful. um, what what was your big project though? So like in your postdoctoral lab. So again, first of all, I bet it's super different, like you said, because it's moving different country,
Yeah.
and there
Mm,
we've had people on the stream who've moved different countries, but it was either U.S. to Canada or Canada to U.S. Right? This seems like an even bigger
mm,
move, not just in terms of geography, but also like different, very different cultures. So, can you
yeah.
comment first? I guess how was the experience of that cross country move or
Mhm.
continent move, right? And then
Yeah.
afterwards, going into like a little bit of what you're working on.
Mhm. Yeah, the move was really, it wasn't stressful because I had really good support from my family as well as my future supervisor. But it was a big shock culturally as well as the temperature was really different. So it was getting, I mean, I had hotel for 1 month, which was booked by supervisor. And then everything was getting closed. Everything was so dark because Danish uh, fall seasons are really dark. It's always raining. So first challenge was to adjust that it gets really early darker here compared to India, which I'm habitual of. But culturally, it was not really shocked because in India we live in a very uh, diverse landscape. So if I move from one state to other, everything is different. Language is different. People wear different clothes. They eat different things. So we are really habitual of these shocks. So
So you already went
culturally,
through that with
that's
your
why
master's and your PhD.
exactly. Yeah, yeah. Both cities had different language. They eat different food. They had a different set of thinking. So that's why Indians are really good immigrants too, because you don't really get shocked directly. You just get buffered very smoothly into new culture, new environment. Yeah. So yeah, the only shock was probably weather, I would say, and the lab was quite international, which was really helpful. And yeah, the institute was really huge, almost 50 PIs working on plants, and it's like a dream come true at that point because coming from the small institute where only 7, 8 PIs works on plant, and then you suddenly exposed to a uh, big institute where everyone talks about plants. Everyone wants to create something new, or people are very healthy, competitive. Yeah,
So it was a like you said a dream come true because of the institute and like how everyone's excited about plant genetics. Everyone's talking about the same topics.
yeah,
Like and presumably the like because it's a big institute, the resources are also very different from the last institute.
different, absolutely. Yeah, it was a different scale. You don't prepare your buffer at all. It's already been prepared by support staff.
So like a world different then in that sense, right? Yeah.
Exactly.
So what
Yeah.
was the research question? And like I guess what was also that got you hooked? Like why when you were applying for labs that you mentioned like it was techniques that you went to the conference and you were like, okay, this is pretty cool, but I'm sure like you
Yeah.
heard the research topic pitched to you beforehand and what your question was going to be. And so what was it
Yeah.
and why did it hook you?
So first of all, I was really excited about the fact that it's a new area to push microproteins, and then Stephen was studying flowering time regulation by microprotein. My personal interest was to move again into crops, but now I study microproteins in crops as my independent project, but then also contribute to other projects those were running in the lab. So I was really lucky that I had this big balance that I could use Arabidopsis as the model, but also study the microproteins and their genomic distribution in crops. So bigger question was actually to establish microproteins as a study material or as a tool because it was a new lab again, new concept. So those early keystone people, they actually contribute more on concept development instead of really finding something bigger. Yeah. And since we were a big lab with a very diverse background, so it was really interesting to learn from someone who is only protein expert, let's say, someone who's bioinformatician, he only does bioinformatics. And then you come into fact that you are a crop expert because you did your PhD on crop. So you had this charm of being a crop guy, but you also want to study process. So bigger questions those I contributed was actually making sure that microproteins could study in crops. So I run a couple of investigations on crop microproteins. For example, one of the tools we developed to identify microproteins from any crop genome using a script, which is now openly accessible. And that was published in G3. And that's how I got to know about Genetic Society. Right.
So is the what crops were you working with, or did it not matter? Like, is the idea of how these proteins work pretty cross organismal, or is it very specific
Exactly.
based on each one?
No, not for me. I mean, uh, there were little struggles. For example, I wanted to study microproteins in barley, and we started writing independent fellowships and grant because my boss had ERC funding, which was for 2 years. So he really encouraged and support my applications to write different local Danish fundings, which didn't really came through. So I got really limited in terms of time I could spend on developing something in Barley. So instead of starting something which would go for 5 years or 4 years, I started looking for a short-term project which was more bioinformatics based.
I see. And so
Yeah, yeah,
that's a transition then, right? Because like with
exactly.
PhD and masters, correct me if
Mm.
I'm wrong, it wasn't so much bioinformatics as much more the hands-on
No,
wet lab approach. So how did that
yeah.
affect you as a researcher? This transition from wet lab to the dry lab approach.
It was actually very exciting because we had really good support from one of my lab colleague who was expert bioinformatician. So discussions with him were really helpful. But it's not just that I was doing bioinformatics, but I was also doing Arabidopsis, which was the exciting point.
So
So
you're
I
now away
helped
from the hexaploid at least.
exactly, yeah. So I helped other colleagues in their projects, and we really published really cool story. One of the good story I would like to tell is one of my colleague who was doing microprotein engineering in rice. So it's really cool because if you look at the overall story, how it works is rice is a tropical crop generally. It flowers when days lengths are long, and in Nordic countries, days lengths are really short. And when days lengths are short, you can keep on growing rice, but they will never flower. If they will never flower, you will never get the grains. So how this project went is Stefan and Tanai. Tanai was a colleague from US. So what she did is they designed a small synthetic microprotein against a bigger protein which controls flowering. So negatively controls flowering. When this small synthetic microprotein goes and bind to bigger protein, it's sequestered into a non-functional complex. So sequestering a negative regulator cause light independent flowering in rice. So how it works is you genetically modify rice by overexpressing a microprotein. Microprotein targets a negative regulator, and then you get rice which can flower independent of light or day length.
Wow. That would be
Yeah,
a benefit to a lot of parts of the world, right, that are unable to
yeah,
grow this food source.
exactly. But since again these are all transgenesis approaches, they are not allowed to get translated very easily into regions like Europe where people are really hesitate of GMOs or those things.
I see. So
Although we can,
you're hitting a
uh,
cultural block almost where you could
exactly.
make this, it could benefit, but so
Yeah.
it is it so the but the locale doesn't want it.
No, yeah, because there's not much acceptance of technology. And again, that's why I was saying in start that now moving to industry, it really helps me see why a lot of discoveries or technologies being developed in university, they don't get into market because it's a big road and there are several blocks and bumps
Hmm.
which need huge amount of support, money and lobbying. Yeah.
Wow, yeah, I didn't even account for the changes. I mean, in the U.S., right, it's not so much food; it's
Hmm.
genetically modified insects to like decrease mosquito populations, let's
Yeah.
say. And
Yeah.
people
Yeah.
don't like mosquitoes, but they also don't like the idea of releasing genetically modified flying
Yeah.
around animals, and so
Mhm.
that's a big challenge. Did you find it? Were you able to? I guess you were the project was to allow these plants to flower at night so you can get seeds without the sun, right? So
Mhm.
you can make food. Were you able to do the transgenesis on it to make this organism, or like what was the end result of it? I suppose.
Yeah, the colleague who was leading the project, she developed the transgenics and then she did a lot of tests in different day length condition and she find that the plants they flower earlier independent of the day length. So it's like a solution to countries like Denmark or in general Nordic countries where if you in future wants to grow rice, although it's not very easy in terms of seeing the temperature and then overall climate, but the climate change is changing and drifting the shift between who can grow what. Yeah.
So
So considering that, it's
yeah, it's bringing us back to the beginning. How you were saying like now you have to account for this like very long term plan that also involves climate
exactly.
change. So I'd be like, well, we
Yeah.
can't grow rice in Denmark because of the winters, but now if you account for
Yeah.
20 years down the line, you're like, well, actually, we might be
Yeah.
able to grow in 20 years down the line. Another
Mhm.
negative
Yeah.
consequence of
Yeah.
climate change, but you're like
Yeah.
plotting out this path for it.
Exactly.
That's really fascinating.
Yeah.
So at this time, you're in the postdoc lab. You're doing these bioinformatic experiments. Presumably, COVID has also hit.
No, it was later. I only spent 2 years in Copenhagen.
Okay, so what
It's
happened
from
next?
yeah 2016 to 18, and I was desperately trying to get some funding because the original grant from my supervisor was running out, which I never got. And then we wrote an application together to one of the funding agency, and then comment we got back was saying this is not the proposal a postdoc should put. It should come from established PI. So Stephen put this proposal again to the same funding agency as a PI, and then the result got due for 6 months. And at some point, my funding was running out, so I decided to find another job, and that's what brought me to Belgium.
And so, is that what you're doing now, or is that something different?
That was my second postdoc
So
in
te-
autophagy.
so oh, and so okay, so it was in immunity. So you got back to immunity in some way. Like you said, you had either plants or immunity. Now we're
Yeah, exactly. Yeah.
was it in plants?
Yeah, because when we were studying Arabidopsis, actually microproteins in general, so I started reading about other ways how post-translationally protein fate is being controlled. So I started seeing a lot of paper about autophagy. It was also very new field which was evolving in plants specifically. There were not too many people who were doing plant autophagy studies. Then I saw advertisement from Belgium about postdoc, and that was also my first application. I applied for the second postdoc, and it hit really well. So I came to Belgium, then I started understanding how autophagy works, but which also brought me to fact that I wanted to use confocal microscopy so desperately. And lab was perfect. We had our own confocal. Everything was so amazing. And then it was another addition into my toolbox, basically, which also really excites me uh, because as a scientist, you want to, as I mentioned, I wanted to experience different science, and it was the perfect that you study first the crop, then bioinformatics, and then development of labs, and now you are studying this autophagy cellular pathway. So it was, yeah, it was a different experience.
Nice. So we have a question in the chat. Do plants have an
Mhm.
active passive immunity? Is it adaptive? Like what even is the plant immune system? And then what component of it were you studying?
I think plants have, I mean, it's really difficult to answer, but if I need to choose from these three words, I would choose adaptive because plant immunity is really complex. It all runs by different receptors, and it's not just autophagy that control immunity, but also other ways. There are proteases, just like animals. Uh, that's how they control pathogenesis. Proteases, or different pathogen effector proteins in plants. It's far complex. Again, it's far complex than being human. Plants are complex. Yeah.
And so what got you excited about particularly the autophagy pathway? Like why did you pick that? Like what brought you to that particular part? And again, I think it's cool that you get ended up in Arabidopsis after all of that. Yeah, we were talking about the different genome composition.
Mhm. Yeah, because autophagy was being studied in Arabidopsis predominantly because it's the best model, or let's say resources are available for our research. And the host lab, they had experience of studying autophagy receptor, which get induced by stress. And how it works is this autophagy receptor get induced, and it use certain cargoes. Those should get eliminated from the cell, and then cargoes through the receptor goes into autophagosome, and then autophagosome goes and then get digested. The content get digested in vacuum. So what excited me about autophagy is again the component that you can actually hijack autophagy. If you think little smart synthetic biology methods, you can choose first to study autophagy, then to identify how you can hijack autophagy to do what you want to do through the plant. That's what a lot of plant pathogen people are doing nowadays. They are using autophagy as one of the first and primary armor against pathogens. And in past, it was more like in background. It's a process running in background in a more silent way. Now it's more vocal. It's more representative because a lot of labs are now focusing on autophagy, especially in plant pathogen field. Although the lab I was working with, they were interested in drought stress and osmotic stress through autophagy because the receptor they were studying it's called TSPO, translocated protein that translocate between Golgi and endoplasmic reticulum membrane, and as a cargo, it get induced by drought stress, abscisic acid. And when abscisic acid is there, then you get stress induced, and then stress cause autophagy activation. And this protein, TSPO, it's first marker of stress, not stress induced autophagy marker. So it was a already established fact that TSPO is autophagy receptor, and my work was there to see if we can identify why TSPO is going into autophagosome and how. So the originally uh, proposed concept was to use proximity labeling, proteomics, where you tag your protein of interest with a ligase. It's called biotin ligase, turbo-ID. And when your protein is tagged with turbo-ID, the tagged protein goes through the autophagy pathway. But since your ligase is very active and going along with the TSPO protein, your protein of interest, everything comes around this complex will get biotilated. And what you do is all the proteins those are being biotilated, you just take affinity purification for biotin and then fetch these protein out and test them on mass spectroscopy. Yeah. So challenge there was to first establish that turbo-ID works in our hands, the affinity purification process. Then also there was challenge to say that the proteins we are patching through this affinity purification are actually protein being interacting or being curated during this TSPO autophagy pathway. Yeah.
So because you were doing a lot of tagging, does that tie back to the microscopy that you were excited about, so that you could see
Oh,
how it was
yeah.
happening?
So yeah, I mean, I personally spent a lot of good time looking at the translocation of protein and then also seeing the microscopic autophagosome-like structures. And because it's more cell biology where you look at the protein relocalization, also protein fade, how it goes starting from Golgi endoplasmic reticulum membrane to vacuole. It was really exciting to look at that small scale, not looking at a bigger genome, but more into small protein, just one protein and one cell at the same time.
That's really cool. We have two
Yeah.
questions in the chat. First is, can plants get viruses? And if so, do you think
Yes.
viruses are alive?
Plants, yes, plants have viruses, and there are really famous examples for plant viruses. And the viruses are alive inside plant.
it's the it's the evergreen question, right? If viruses are alive.
Exactly. Yeah, yeah,
There's
definitely. Yeah.
never a good answer.
Yeah. And plants are, I mean, viruses are alive inside plant, and there is a dedicated pathway for viral autophagy where virus infection activates autophagy, and then there are groups those are working with virology of autophagy degradation.
I see. So that was one of the
So,
stressors that
yeah,
you could use to induce that pathway that you were studying.
yeah.
Nice. And the second question is from your postdoc work. What was one of the most unexpected results you've got out of your research? How did that result change how you were looking at what you were studying?
So one of the unexpected result was actually seeing a cup-shaped structure during this microscopy imaging. So I was supervising a master student within my project, and we used to spend a lot of time on microscope. So one day I mean, it was late evening, so I asked him, "Do you want to get beer?" Yeah, yeah, let's get beer. He's Belgian. He's really cool guy, and he really likes beer. So after finishing a long day on microscopic room, you really want to get out, sit in balcony because our office had balcony, and then drink a cool beer and go home with satisfied result. So I asked him, okay, I will go and fetch beer. Why don't you keep looking at the microscope? I mean, he was looking at the sections we prepared. And then suddenly I got call. He was saying, I saw something very amazing. Because what we were trying is trying to see if we keep cell non-stressed. And if we induce stress using different agent, for example, ABA or a salt stress, do we see change in the aggregates we are seeing? So he called me and said, I don't want beer anymore. I'm already very excited to see something. So I went to the microscopic room and we saw in one of the stress condition, we started seeing a cup-like structure where you see a hand more like this, and then you see aggregate being stuck inside. Yeah, and then suddenly there was a bubbling, and then this thing got out, and then started going around the cell because it's an endocytic pathway, and if you use right marker, you could locate. For instance, in the same cell, you can use M-cherry tag to one of the ER markers, so you have all endoplasmic reticulum visualized. You can tag your protein with one of the marker, for example, M-fucoase, and then you can use the endocytic pathway marker. So three different colors and tagging different subcellular environments, and then you see this dynamic running. And which in dark room, if you look at this big screen, and everything looks so out of the world.
So these are in like
It's like a
live functioning cells that you're able to
exactly.
make different
Yeah,
parts glow and actually see the process
yeah.
happening in real time.
See the process exactly in just one set of cells. Yeah. So we started seeing these cup-like structure, and he questioned me, is it like origin of autophagosome that it's hand-like structure which is trying to trap protein inside so that it get close? And we were really wondered about it. Then we started using more markers, the autophagosome marker, for example, and then we really saw that when autophagy like structure starts, it actually starts more like a, a curved hand, and then suddenly you see closing and then maturing. But again, our microscope was really old, so we couldn't really confirm everything. But that moment when we started seeing those structure, it was really amazing. Yeah, I still have those movies with me. Sometime when I feel out of the way from this whole corporate business, I really sit down and look at all those images.
Yeah, we'd love to see that and share it with the community if
Uh,
ever you have access to
mm,
them. Yeah. Um,
uh,
so that is really cool. So what was the end result of your postdoc work in terms of like the question that you answered?
so that was the project I just explained. It was just one part because I was also doing another crop project because I'm a crop guy. I always like to keep this balance. So our lab had a project about biofuel crop. It's called camelina, and camelina is really famous for biofuel because it has really good amount of oil in seed, and it's a future ready crop because it really could grow in drought environments. It doesn't need a lot of water and too much of nutrients in soil. So what my boss proposed is that let's try to do more translation part using the autophagy and autophagy receptor we were studying TSPO. So what I tried is we try to see when in natural condition TSPO expressed when seed is really mature. What we did is we used a synthetic promoter and then we expressed TSPO protein during seed development, but very early stage. So since it's a sensor of stress, the TSPO protein, and you express it at the end of seed maturation when seed is already dry. And if you bring this protein earlier when seed is really active, accumulating all the metabolites and storing fats and carbohydrates and protein, how does it change? So we early expressed this protein, synthetic protein, the TSPO protein using synthetic promoter. And when we did metabolic assays, metabolic profiling of these seeds using a platform with our collaborators, we saw that there was a big shift in total oil content of seed, and which got us question like how appearing early this protein is changing all the metabolic landscape for seed. And we saw at least twofold more omega-3 fatty acid in the seed. So then I went into the way and we did some RNA-seq during seed development process. We identified that when a protein appears around 18 days after flowering of seedage, then it changed a sense of stress in seed. And when seeds see that there is a stress-induced protein present here, it means that environment has changed and seed should get matured earlier. So it's a panic state, and how I call it is genetic priming. So you are sensing or you are forcing your seed to sense a stress which is not there, and seed start biosynthesizing all the storage product very quick. And then since it's a oil seed crop, so all the focus went into the oil content increase. And when seed mature, because it's a panic stack, it stores a lot of seed because considering physiologically down the lane, seed needs to grow. That's the purpose of seed. And if you are not matured enough and you don't store uh, energy for seed, the seed won't germinate. So it's like a biohacking the biology of seed, but to do something which seed usually won't do. So what we develop in the end is we increase up to 62% of oil content in seed for this crop.
And that's also why you called it, and it's more of an adaptive response, like you were
Exactly.
saying, because you're able to
The
prime
adaptive
it and then
response. Yeah,
prepare for that a second induction of the stressor if you so
yeah,
choose.
yeah.
Nice.
Yeah.
So
And that's why I was saying plants are really cool if you look at the plasticity because they need to make immediate decision to survive next generation. Yeah,
yes, there's that transgenerational effect as well.
exactly.
Yeah,
Yeah.
it's a beautiful system.
Mm,
So during
mm,
your postdoc time, you also joined the early career leadership program of the Genetic Society of
yeah.
America. And could
Mm,
you tell us a little bit about that experience and the projects that you led there, and how that kind of
mm,
ties in to your original passion of, I would argue, science for everyone, because of like, even the scientists with the farmers at the right, like it's kind of tying back, and I feel like you have pushed that in a lot of your endeavors. You've even started a Twitter page for
yeah.
scientists and like plant scientists in India to like communicate, the works of the plant biology. So can you tell us a little bit
Mm,
about what you did at the GSA ECLP program, and how that ties into your overall, I guess, one of your overall
mm,
mission statements of that science
yeah,
communication?
definitely. Yeah. I think it was during my early postdoc time when I started realizing that the community overall and then peer support has a big impact on your career decisions as well as your personal growth. And when I published this paper with G3, I started looking at the blog which GSA was running, and there were a lot of resources actually. And when I subscribed to the resources and newsletter, I saw ECLP advertisement. So I was not really sure if I wanted to do that, but I had experience of working for American Society of Plant Biologists as a Plante Fellow, which was really amazing experience for me. So I thought I should give it a chance, and I was lucky that I got this opportunity, and not just being a member, but they found me suitable to co-chair the subcommittee for community and membership. So, and early, since it was a big commitment, so I felt a bit overwhelmed by the fact that now you need to organize meetings, manage some projects, or follow certain progresses and those things. But with the time, I realized that I'm learning some skills. Those were very untouched, and untouched in sense that I could really write an agenda, and it was very professional way of writing agenda because the co-chair I had with me, she was really, really professional. She was really amazing. And these conversations we had within the community, they started giving me this vocabulary, which I was not really aware of. For example, I recently, when I joined this new job, we had an induction period where we spent time off-site and we talk to our colleagues about it. So during GSA ECLP training, we had this leadership training, then inclusive and inclusion and diversity training. So those training gave me this right word, right vocabulary, right direction, how to think, what should I read to develop myself as a person, but also as a leader. And also, I got more aware, how should I talk to my colleagues. And I also start seeing that there is a lot of opportunity to really advocate about what is going wrong in academic research in general. Because the GSA provided this safe space where people were really talking about issues those you really can't talk outside. And why you can't talk outside? Because first of all, people will start judging you. And then beyond judgment is that you could get consequences of saying those things in open, yeah, or on social platforms. So I got really hooked to these conversations, and especially the mentors we had for subcommittee as well as for the steering committee. Those were really amazing people. I mean, the perspective they put into discussion, it's a different way of looking at things. And that was also time when I started seeing, talking to my other peers, the fact that there is a leadership vacuum. And often the leadership vacuum doesn't really replicate into leadership, but also mentorship vacuum, that there are certain things you can talk to your professional mentor, seeing insecurity of yourself or your own mentor's insecurity, which you can really take your question to one of your GSA meeting and then shoot it to one of your mentor, which is not your formal mentor, but mentor from GSA. And those questions, those discussions, they really open mind. They really helped me through a couple of difficult periods professionally, I would say. And yeah, that was also time when I I conceived this idea to start a leadership dialogue series because uh, although we really got formal training sponsored by GSA for leadership, which really helped me look at the way of leadership should go or training should be. But it's not a dialogue which is going openly that PI should also be a leader. They should not just focus on their own career, saying, I had this grant, I want to hire a postdoc, which should work, publish these papers, and then the ultimate benefit goes to the PI because the guy who, or let's say the associate who's working on project will only get benefited scientifically by publication. But in general, there is no personal growth unless you are surrounded by good peers or a micro community where you get enriched, you get chance to discuss those things, which I was really, which I was really fortunate to have through GSA. Yeah.
And
So
that's
ECLP is really amazing. Yeah, for that.
and that project you're helping lead now, and the first series will be up on the YouTube page and not just too long, right?
Yeah, definitely. Yeah, we are really looking forward to that. We finished recording for first session, and we have lined up really amazing speakers for that series. Yeah,
Yeah, that's
it
going to
will
be
it
really
will really.
exciting.
I'm really looking for the ripple effect, how we start conversation about the necessity of good leadership in science, and then this should break this taboo. Especially one of our guests during informal meeting mentioned that if you are not a good leader, you should not leave your job or say, see, I'm not a good leader. But you should definitely work on yourself and say, don't worry, I will work on that part or I will improve myself, which I have seen a lot of new PIs are now doing more consciously that they just don't want to be scientists, they want to be effective leader, communicator.
And
Yeah,
hopefully this dialogue series will further open that discussion and push that discussion forward.
absolutely.
Yeah.
Yeah, that's what I'm hoping for.
No,
That's what our whole committee is hoping for, actually.
yeah,
Yeah.
that's no. I'm really really excited, and again, that's a huge project to lead. And like you were saying, it's something that you kind of have to be quiet about, but you're kind of just I don't care. I'm going to put this at the forefront and try to build a difference in the
Mhm.
scientific community. I think
Yeah.
that's a very
Mhm.
laudable effort, and thank you for leading that,
Yeah.
Kaushal.
Yeah,
Um,
thanks a lot for supporting us.
oh, we actually had a question earlier that I think kind of applies now.
Mhm.
Is that after
Yeah,
you finish your postdoc, you transitioned to a different type of job, as we were talking about at the beginning, more of that industrial look to it. So the question earlier was,
mhm.
how does one go from the specificity of a PhD thesis, i.e., studying regenerative properties of planarian, or you studying autophagy in a particular pathway, or making a mutant in this one particular line of species that has
Yeah.
hexaploid, right, to something like in the private sector where you maybe want possibly a bit more of a general, broad approach. So
Mhm.
how does one go through that transition, and how did you identify the proper quote unquote location for you to land?
Mhm. I think I mean, how I see always is when you are in your current job, you should always look for people who are on your next job. And when I say your next job is like you could imagine yourself working as a PI in academic environment or a scientist in industrial environment. But what we often do is we don't really get consciously trained for fact that before moving to your next job, you should be able to train yourself in your current job that prepare you for the next job. So as I mentioned, my supervisors were really helpful in identifying my personal strength. And more than supervisor, it were actually people I mentored, those who helped me identifying that beyond just doing science, I could also go and push other boundaries. For example, this leadership series, which we just discussed. And uh, and actually, if you look at the profile that industrial scientists need, you need to think as a scientist, but you need to work as a manager. You need to know how to negotiate between three different parties. And also at the same time, you need to know that what you are saying is actually going on the record every time. So I felt the recruitment in industries are more oriented on a person and then skills. While in academia, when you are hiring a postdoc, you want to hire first a scientist who is expert in XYZ technology, and then you look at the perspective that how good is he or she or them in terms of being human or being a good colleague and those things. So as I mentioned that I had really good peers and colleagues, those who really shaped me. And while shaping, we also had these discussions that one of my good friend who was colleague once, and she always says that you should not limit yourself into just doing science, but bring more out of yourself. But to bring more out of yourself, you really need to train yourself first. And that's the part which I felt really helped me because looking forward to the next role, I always try to train myself in the current role so that I should be ready for the next role. Yeah. And in industry, it's really different. Different in sense that you need to be really agile and you need to have this mind switch ready. As I mentioned that sometimes I'm sitting in a meeting where we are talking digitalization of database or using AI in certain research project. And then suddenly you are talking about how to optimize a protein assay or identifying the issue that a certain machine is not working and you are expert of that. Now you need to help us. So, and then at the same time, you are talking to regulatory people or IP people saying, we have this technique, we want to need to IP protection about it. And then you have certain project ready or product ready. You need to negotiate with the regulators. How are we going to release or develop a regulatory document about it? So it's it's just you need to prepare yourself. And when we apply for the next job, we also need to see if we really fit into the role. Because sometimes if we don't fit into the role, we get recruited, and then it gets really tough time. Yeah,
So it was
and
very
not every industry is ready to act you. I mean, first take you and then create opportunity based on your expertise. and that's where most people make mistake in choosing the right role.
We have a question from the chat, and I don't know about the term, but is the question is the private sector more of an agile work style than academia? I'm not familiar with the concept of agile work, but maybe you are, Kaushal.
Yeah, I am. Yeah, so agility is more like doing different tasks and then switching from one project to other based on need of project. Because, for instance, we have, I mean, we have agile working way, which was actually pioneered by Google first. So in our office, we have projects, and then each project recruit an agile team. And how agile team works is project manager decide I want these expertise in the in my team, and then experts will then come together in agile team and contribute to just one task. But as an expert, you could be part of multiple agile team. So, for instance, you will be able to contribute uh, to one software development, one bioassay development, other genetic mutation development, then other pipeline development or a negotiation development. So these are all like a different roles being assigned to you through different projects, but it's you just one person contributing your expertise into multiple agile teams. So it's a really different way than academia. And if I compare to academia, you once you start your job, you have been given a project. Max you could do is you can collaborate with your colleague or your neighboring lab or one of the collaborator of your boss saying, I feel a little bored about this project. Could I start a side project? But the side project will also be about the same subsidiary of your main project. So it and after doing this agile way, I feel more alive because I don't focus on just one thing every day. On my task list for the whole day, I have really diverse topics.
So I like that
Yeah,
you said you feel more alive. That's a cool way of
yeah,
phrasing it. Is there
yeah.
any other pointers that you would give to people that are considering the potential of going into industry, that things along the lines of, like, you felt more alive, and like other ways that you felt it was perhaps beneficial to not just your like future career, but also like your mental health and like how you end up
Mhm.
living life, because you also have a family in the background, you're in a different country,
Yeah,
you also presumably have the hobbies, like I see all the plants in the background,
yeah,
right? Like balancing all these things and picking the right thing for you. I know it's going to be different for everyone, but like,
mhm,
what were your
yeah,
really important values that led you to this decision point of, like, this might be the path that I want to take?
yeah. I think in terms of work-life balance, I always had one, starting from my PhD, because a lot of my colleagues were spending time in lab, but I believe spending time in lab and then being really productive are two different things. So, and I always put forward fact that you are you are a human first, and then you are a professional. And being human, you have certain needs. You need connections, you need relationships, you need to maintain your health and also the mental health, of course. So from starting, I always had agenda that I want to do sports almost every day. And if I'm not doing sport, I need to read a book. So how I do it is I always put my personal agenda in the day, which actually then help me to adjust everything else around the day. So how I'm seeing it that I must go for a run today. So it will help me and keep me motivated for the fact that finish all these tasks, the professional task I have assigned to myself, so that I may prioritize that I will go for a run today. And then other way is like I want to cook really delicious meal today with my wife in kitchen. So that's my main objective for whole day. And then everything else professionally will fit around it. So what it does is I'm excited for my evening with my family, and that's why I'm more motivated to finish the task. Those are more professional and without being involved and without being mentally affected by them, the failure or the success for professional task. And that's how I have always balanced it. So in addition to that, I also like my personal growth, and I felt that personal growth often comes from the volunteering and then non-professional participation. For example, in micro communities, or in programs like ECLP, or running an initiative, or volunteering to something which benefit the community in or the society in general. And if you are really that open-minded and you want to talk, you want to bring the changes, then you can also keep that as a center of motivation, and then fit your schedule around it. Because often that our whatever motivated you are to do science, to be a manager, or to do whatever in your personal life, at some point you will feel like you have lost this vision, or you are feeling out of space. And to come into outer space, you need those priorities: life, family, relationships, and then your personal core values. Core values could be society, making a change, benefiting someone. So, for example, in last 6 months, almost every weekend, I have contributed 1 hour in talking to someone who has approached me through LinkedIn or Twitter. And it really helped me looking at the perspective which people are sharing with me. And it also helped me in seeing that how or what kind of changes we need in the classical PhD or postdoc training. Yeah. So fitting my agenda now around the volunteering part I'm doing every Saturday around 1 hour, it it gives me a perspective again that I need to help this person, I need to talk to this person, and then I schedule everything around it. So it's all about personal core values, and again, I always say that put your life on priority first, and then you will see everything makes sense around it.
I like that
Yeah.
mantra a lot. And actually, I wanted to
Mm.
highlight some of your works, in particular in for the community. So one, this is your website, which I wanted to link again for everybody in the chat. If y'all are interested, we'll pin it so folks can check it out. I saw and read you had started this India Plant Science Twitter page as
Yeah,
well, and that was another means of like almost this mentorship, right? Of like disseminating information
exactly.
outside of science. Can you
Yeah,
tell us a little bit about this initiative? There's also a Facebook page that you're also running about
yeah.
sciences. Can you tell us a little bit about these?
So the Facebook page actually came first when I was young PhD, and again I had a lot of time left because I was finishing my day task very early, and then when you have time left, and I didn't have family, I was single then, so I started reading, and I like reading new discoveries, new documents, and new papers coming up. So I started this Facebook page just to make awareness that there are a lot of things happening, and there are a lot of new technologies are coming up in agriculture, biotechnology, and genetic area. And when this page was actually at peak at some point, so I started getting requests from people who wanted to invite me to talk about like a like a club seminar or something, which was really cool because I really like talking about new discoveries in plant science. So then, when the Twitter was in boom, and at some point I started seeing the representation was really low for Indian plant science. So I talked to my friends, and friends those are already in India. They are running their own labs, and they really appreciate the fact that yes, we need a platform, a common platform where people could tag our page and we retweet their discoveries or their publications or the advertisement they have for workshop, for instance, which should benefit the early career scientists because we need to provide the voice to people. And in a bigger country like India, running a small micro community is really difficult. So it's just an initiative to give the voice, create a platform or a center where people could come back and look for references or resources.
So it's almost like a community resource board that you're curating
Yeah,
for
mostly.
scientists and non-scientists alike.
Yeah. Mm.
Yeah, it's an amazing goal, Kaushal. Like it's almost pulling back to your very origins, right? As like, farming with your family and learning from the
Mm.
scientists, and then in your master's
Yeah.
work, right, you were giving back as the scientist to the farmers, and now it's like you're just giving back to an even broader community. And I think it's just such
Mm.
a remarkable journey that you've gone through, and like kept
Yeah.
those values the whole time, which is, as you said, people can lose sight of that quite easily.
Exactly. Yeah. Yeah. I mean, professional motivations are often inspired by success, and when you don't get success very easily, you lose the sight, and that's the time when we should look back to the core values to see why you came this path. Because for my personal objective, my journey starting from a small village working on farm to so far is it's already a big achievement, I would say. But it's also fact that it takes a village to raise someone, which is mentors, then peers and friends. So, so to give back to community, it's really important. And everyone should be able to speak up if there is a wrong thing happening, or if they want to help someone. Because I heard a lot that XYZ write email to someone for help and they never reply, which is very common in academia, and I really don't like it. You should give back as much as you can. If
I
not financially, at least you can spare your time with someone in need.
and that's again why you started the leadership dialogue series, right? Is to be able to
Exactly.
encourage
Yeah,
folks to give back.
yeah, yeah,
Yeah, it's
yeah.
it. You're amazing, Kaushal. That's all I can tell you. It's
I mean,
someone fighting
I mean,
for.
yeah, this community is amazing. It's just that we need to be more open and more vulnerable to speak up and share personal experiences, which people are not often doing. And it got me worried that if you want to build a sustainable community, you need to be able to share and speak up very well. Yeah.
So while folks gather any last questions that they have, do you have any parting thoughts or wisdom or something that you'd like to share with folks? Again, we're going to plug all your socials at the end, but something else that you feel
Uh,
like we haven't yet talked about that is really important to you, or that you feel like should be important that people learn and hear about.
mm, I think one of the burning thought I often learn from the wise people I have privileged to talk to, and they often say that don't compare your journey to someone else because everyone has a different starting point. And also in the end, that your uh, your overall path is actually very individual. So frame, trace, and make best of your path without being affected that someone what other person is doing or what path they are following. You don't need to really follow anything. And I believe in this fact that Gautam Buddha, when he got enlightened, he said that there are ways I can answer your questions, but there is your own path which you need to find. And so it's everything starts from this conscious fact that you need to find your own path, and you just stuck with to your core values, and that's it. Comparing yourself with others in any mean, any scale is not fair. And that's what brings more sorrow to people, seeing that, oh, I should have been done that, or I should have followed that path. So, yeah.
I like that a lot.
I don't know if it is a wisdom, but I think it's philosophy I learned from more wiser people. Yeah.
Oh, it's a good philosophy to have, I think. Nelf
Yeah.
says in the chat, I wonder what fantastic science insight have not been discovered because of the lack of access. And so hopefully with your approach, like we'd break down those walls and people will have more
Mm.
access, or even within the scientific community, right? More of a support system where you wouldn't lose
Yeah.
people in
Mm.
certain positions because of just how the system is built.
Yeah.
We have a question from the dinosaur Dave. Final question for you. What is your favorite dinosaur?
T-Rex. I have seen the real skeleton of only T-Rex so far.
The what is it?
T-Rex. I was saying
Oh, the
that
T Rex?
or probably the No, I think that's the best one. I like it's small in comparison to other dinosaurs in terms of size, but also it's very powerful. And yeah,
I have a silly follow up question to that as well. What's your favorite plant? As a plant scientist
ah,
throughout your whole life, and I see there's a bunch of them in the background, do you have a favorite plant?
no, I think I like staring at the. Uh, I have a favorite tree. I will find the scientific name for that. Let me quickly. Uh, Banyan tree. Okay.
The what kind of plant?
I I like banyan tree.
Okay, what kind of tree is that?
It's like it's one of the biggest tree.
Okay,
I will paste a picture. I mean, I'm personally very hooked to this tree because of a certain reason. I mean, it has a really big value in Indian culture, but also philosophically that the village where I grew up, my farms were surrounded by two of them. They are like really old trees, and I felt sometimes that life often revolves around trees. If you put one, then I will probably wouldn't be able to make picture, but I think everyone can Google it.
okay. I'm gonna show this to folks.
Yeah.
Let's take a quick look.
Mhm.
Oh wow! All right, these are absolutely
Yeah,
beautiful. I could like that would
exactly.
be also like I feel like a kid's dream to climb on something like that. I want to climb on it now as an adult.
Yeah, sure. But compared to the crops and plants, I like trees more. I could sit in front of a tree and then look at it for like really long time. Just appreciate beauty. Oh, how beautiful a tree could be.
I understand. I also have a favorite tree. Like I like to just
Mm.
sit outside the house and look at my favorite tree, and it's just it brings me so much peace, and I really enjoy just
Yeah,
it fills you up, right? Like there's some kind of spiritual
of course.
connection that you can have with a tree.
Yeah. Mm.
No, I like these are beautiful.
Yeah. Mm.
So I think with that, Kaushal, thank you so very much for chatting with us today. We'd like
Yeah.
to shout out all your socials that people can go follow you. Do you have any upcoming events or anything that you would like to plug before we go?
No, I think I would really look forward to the leadership YouTube series that will come up soon.
Perfect. I'll link the YouTube
And
in the chat
yeah,
as well. There's our YouTube
yeah,
page, and it'll
looking
be on
forward
the
for it
GSA
being there.
YouTube hopefully soon.
Yeah,
Because the first video you already have done, we're just in the process of
yeah,
processing it, and then we'll have it uploaded. So make sure y'all follow that YouTube page as well for more of Kaushal's work. Um, Thank you so very much again, taking your time, chatting with us about your journey, which is again absolutely incredible from the beginning of the farming to like talking with the scientist to back again as
yeah,
being the scientist, the farmer, all the insights you've shown us about the what you've been working on, the family moving as well, the crazy timelines of like
yeah,
defending your thesis, getting in the car, moving to a different country the next day. It's
yeah,
it's been a beautiful ride, and we can't wait to see where you go. Thank you so very much,
yeah.
Kaushal. As
Thanks a
folks,
lot for
yeah, go ahead.
no, thanks a lot for being amazing host and GSA usually is really amazing. Everything GSA does is for community.